Okay. All of you can see my screen now?
Yes, sir.
Okay. Okay, guys. We are talking about PCR RFLP, right? Now, PCR RFLP, just to simplify the entire technique, what people have done is, like, instead of getting along with the entire DNA and breaking that into pieces and then running onto the gel and using the radioactive technique, they initially used to amplify a particular segment of the gene. Now, what did I tell you? You already knew here that at this particular point, there exists polymorphism, right? At this particular point, there exists polymorphism. So they used to amplify a fragment adjacent to this particular known site, right? And then let us say that fragment can have 500 base pair, right? Amplicon or the amplification product. And they used to amplify that. Now you have this 500 base pair fragment. And if the site is present within the fragment, let us say the site is present at the 300th base pair, right? And this is the 200th base pair. And if it is present at the 300th base pair, you'll be having two segments, 300 and 200, when run on a gel, right? Whereas when the site is not present, you'll be having a 500 base pair fragment. That is what is PCR RFLP. Is that clear? Hopefully it is clear. We are just telling you the same old story here, like having got polymorphism at a particular spot in a gene, you are going to amplify the adjacent fragment of that particular polymorphic site, and then using primers, and then digest it with the restriction enzyme. What do? You initially amplify that region and then digest it. So you have a 500 base pair fragment here, and then digest this with RE enzymes. So first is nothing but a PCR amplification. And then after that, what you do is you RE digest the fragment. You RE digest the fragment. That is what you do. Now, this is all bookish. You can find it in any book and you can do it. Now, the most important thing about PCR RFLP. Now, what I say is, this is a marker that is said to be codominant. Codominant. Now, what does that mean? Right? When you say a marker to be dominant, or we'll talk about dominant markers also later on. But first, initially try to understand what is this codominant marker. Now, when you say a marker is codominant, it means, see, when you're running a gel like this, if in an individual, you have two chromosomes, right? One is maternal and the other one is paternal. In one of the chromosomes, when you call it, there is a polymorphism existing at the site. And also in the other chromosome, there exists polymorphism. Now, this is individual A, right? For this individual, if there exists polymorphism, what happens? You are going to have a band at this length and at this length, and this is called as 300 base pairs, and this is called as, this is around 200 base pairs, right? Now you have two fragments that are amplified. Now, let's say in this individual, in the second individual, if you have, let us say, polymorphism in one site and you don't have polymorphism in the other site, giving rise to what? Giving rise to a second allele. Then what do you have? You will be having, for sure, when you are digesting the DNA from this particular DNA, you will be having both these segments. 500 will be amplified and will be cut. But also you will be having a segment that is around 500 base pairs. Means by using polymorphism, this corresponds to allele A and this corresponds to allele B, where allele B does not contain the restriction site. RE site is absent here. And when the restriction site is absent, we'll be having a 500 base pair fragment here. So the existence of both the alleles in the population in an individual can be identified. Means here what? Both the alleles A and B are identified simultaneously by using the technique called as PCR RFLP. So this is therefore called as codominant. Means A can also be detected and B can also be detected. Is that okay? And similarly, you have an individual B, let us say. You have an individual B having B and B. B and B, what we have taken, the restriction site is absent in B. That's what we have taken, right? If it is absent in B, what will you have? If you are talking about a third individual, we'll just extend this gel here. We'll just extend this gel here. So if this is the third individual, what will you have? Have an RE site. RE site is absent in B. So you'll be having only a fragment here. So this is individual 1, individual 2, individual 3. Individual 1 is homozygous for the presence of the restriction site. Individual B is having the restriction site in one and absent in the other, whereas individual C will not be having the restriction site at all. So this process of identifying all the alleles together is called as codominant. Is that clear? Called as codominant in nature. So this marker is said to be codominant. Okay. Now we'll go to the second technique that is called as RAPD. What is it? The second one is called as RAPD. Rapid Amplified Polymorphic DNA. No, I don't know. This is not used anywhere. People are still using RFLP, but RAPD is not used. This was given by Williams. This was given by Williams in forgot the year, guys. I think it is 1990, but I'll correct myself tomorrow or day after. Sunday, I'm not taking classes, guys. We'll take it on Janmashtami din to nahi hoga. I'll let you know. Right. Even if we cannot finish this topic in the 2 classes, we'll take it tomorrow. We'll do it. We'll finish this off. See, now what is this RAP? See, most of you, I'm very sure most of you don't understand this. Right? You definitely don't understand this. I'm very sure because we are not doing it, and no one does it. No one does it in the current scientific world because this is not repetitive at all. This technique, the technique is not repetitive at all. This is not repetitive at all. Okay. Now, the most important thing. RAPD, normally for a PCR amplification, right, you need 2 primers. You need 2 primers. But for RAPD, you need a single primer. Now, don't ask me, with a single primer, how are you amplifying, right? You need a single primer. Now, what does this mean? The primers are so designed, listen to me very carefully. When you have a DNA strand like this, right, complementary strand, 5' and 3' here, and here you have a 5' and 3' here. The primers are so designed that there is a segment here in this direction that can bind to your primer. And also there is a segment that is there in the reverse direction for the same primer to bind in this direction. Is that clear? So they are defined primers already, right? Now, once again, I'm repeating this. The primers are so designed that you have a segment of DNA here that is just in the reverse direction in the opposite strand. And when you have it in the reverse direction, this primer, the single primer that binds to your forward strand, let's assume this to be forward and assume this to be reverse, the one that binds to the forward strand would again bind to your reverse strand also in the reverse direction, because you already know that the primers are designed in such a way for those regions that are repeated, that are reversed in the opposite strand, right? Now, how does polymorphism exist now? We're talking about polymorphism. How does this exist? Now, let us see. You have this strand here. You have this strand here, right? Now, this is 5 prime, and this is 3 prime, and this is 3 prime, and this is 5 prime. Now, what are you using? You are using one particular primer. Now, let us say this is binding here, this is binding here, right? This is also binding here, right? And this is also binding here. This is also, okay, not to confuse you so much, but this is also binding here. Now, what are the different amplicons that you get? One amplicon is between these two. This is the amplicon. One amplicon can be between these two as well. So this is the amplicon size, second one. One amplicon can be between these two as well, right? You'll be getting this one. And one amplicon can be between this and this, right? As well as you can have an amplicon between this and this. So you're using a single primer. So how many fragments are you identifying here? Just listen to this. How many fragments are you identifying here? You are identifying 5 different fragments that can be amplified with one single primer. Now, 1, 2, 3, 4, and 5 fragments. Now listen to me. Having got this, how does polymorphism exist now? Now this is, let us say, an individual A, right? Now we'll take an individual B here. Again, draw the same stuff. Right? This is 5 prime and this is 3 prime. And this is 3 prime and this is 5 prime. Right? You have, again, got amplified. You are getting amplified this particular region, same primer here. Same primer here. Now, per se, in this particular individual, there is no binding site here for the primer. So this primer will not bind. And similarly, let us say you have a binding site here. Now, how many fragments do you have for this particular individual? You have this fragment. You don't have this fragment. The first one will be there. Second one will be absent. Third one. What is the third one that we decided? Third one will be also absent. And then you'll be having the fourth one and the fifth one as well. Right? Got it? So, what happens? So, it depends on whether there exists polymorphism at those sites for those sites where the primer has to bind. Now, having known this, this is called as a dominant marker. Why do you call this as a dominant marker? Now, why is this called as a dominant marker? You should understand. Now, listen to me. What? At a particular site, if you are able to identify the existence of a heterozygote as well as the homozygote, then you call it as a codominant marker. But here, what is happening? At this particular site, it is either here it is present, it is present. At this particular site, it is present and absent. So it is only presence or absence. Is what? It's either presence or absence of a particular band. Right? There is no question of anything intermediate between. Presence is dominant, absence is recessive at a particular location. So you don't have this band, so it's either present, it's absent here and it's present here. And that is the reason why you call this as a dominant marker. Call it as a dominant marker. Here it's clear. Anyone give me a heads up? Or am I teaching simply to no one listening to the class, expecting that I would share the recording? All of you are listening?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay, guys.
Yes, sir.
Right. So this is what is RAPD. This is what is RAPD. Now, the problem with RAPD is RAPD amplification normally is done at 37 degrees centigrade. What? At 37 degrees centigrade. And when you do it at 37 degrees centigrade, what happens? The repetitiveness of this particular technique, the repetitive nature is quite poor. So this is not reliable at all. And so people left this technique. I just wanted you guys to know about this technique and also to differentiate between the dominant and the codominant markers. I wanted you guys to understand this. So we have finished two techniques among the anonymous markers. And I told you what anonymous markers are. And so here, the next comes the mini satellite and the microsatellite markers. Mini satellite markers. Mini satellite was given by Jeffries. Again, I forgot the year. Probably it's 1985. I'll let you know. I know it's in 80s, but 1985. Sorry, guys, I forgot. But anyways, the year I'll let you know. Right? Now, see, a mini satellite is nothing but a tandem repeat that can repeat from 6 to 60 base pair repeats. Means when you say it is a tandem repeat, what does it mean? When you say it is a tandem repeat, what does it mean?
Repeating one after another.
So you have this here. So you have this here. You have this here. You have this here. Repeating one after the other. Right? Now, what you should understand is, anyways, we talked about mini satellites and we said like most of the mini satellites are present in the telomeric region, right? In the telomeric region. That's what we discussed. That's fine. But here, what is the reason for polymorphism? Now, when you talk about a box here, for example, if I have boxed something here, it means, right, this particular repeat can be 6 to 60 base pairs, means one particular type, not this cannot be like, let us say this is 6 base pair, this will again be 6, this will again be 6. Don't expect that if this is 6, this can be 7 or 10 or 20. The same repeat unit is repeated one after the other. Now the polymorphism is basically because of the number of repeats. The number of repeats. For example, in individual one, like for example, at this particular spot or at this particular region in the population, if there are 20 repeats, in some other individual taken from the population at the same region can have around, let us say, 24 repeats. Is that clear? Now what happens, just because there is a difference in 4 repeats, when you run it on the gel, there will be a difference in the segment running through the gel, and you can identify the polymorphism. And no two individuals in the population will be having the similar kind of repeats on the chromosomes, and this is popularly used for DNA fingerprinting. DNA fingerprinting. I'll just explain what this is, but just remember DNA fingerprinting. Now, people call this as variable number of tandem repeats and all like that. You can just look into, but that's fine. But okay, now what is it? How is polymorphism identified? And now what do we have? See, hopefully all of you are there. So what do we have now? Let's take one. Okay, let's take individual one. Individual one. And now you have run that DNA on the gel. You have run that DNA on the gel. And then you run that DNA on the gel. Again, what happens here? You're expecting that you're digesting with restriction enzyme. And again, you're running it on the gel. You expect that there'll be a smear all along, right? Now what happens? These mini satellites are present elsewhere in the genome as well, right? Distributed across. Now, when you're doing this, you normally use a probe that corresponds to your mini satellites. Probe that corresponds to your mini satellites. Now, what happens? For example, in an individual at a particular locus, we are talking about at a particular locus. If this goes and binds, let us say you have a strong band at this particular point, just because it has got 20 repeats. Now, for an individual at the same locus again, what happens is a second individual, if you're looking at, you're running it on the gel, right? Now what happens, if this individual has got 24 repeats, it's the same locus again, mind you. But just because it has got 24 repeats, the band that ought to be present at the same spot at 20, if it has got 20 repeats, will now be a bit at a higher level. Will be present here at this particular locus, right? So this is the way that the actual difference, even at the same locus, just because of the number of repeats that you are using, right? Since you have used a probe, you can differentiate the number of, you can differentiate the kind of bands you get mainly because of the difference in the number of repeats. That's fine. Now, what next? Now, this can be present elsewhere in the genome as well. So let us say for individual 1, this is present elsewhere in the genome, and it has got, let us say, 30 repeats, right? So this will be around here somewhere, right? And this is also present in this individual, and it has got only, instead of 30 repeats somewhere else in the genome, let us say it has got 20 repeats or 20 repeats or something like, not confused, we'll put 15 repeats. So we'll have a 15 repeat band here. So likewise, what happens? Mini satellites now will become multi-locus. Why multi-locus? Because you are identifying at different loci. Is that okay? Understood, right? Multi-locus, multi-allelic, multi-allelic. What does this mean? Means at this particular point, for example, at the same locus that we are considering, listen to this very carefully, guys. I'm telling you, right? Very important, and you should understand this. Why is it multi-locus? Can be present at several places in the genome, but at the same locus alone, it can be multi-allelic. Means one individual can have 20, the other individual can have 24, some other individual can have 26, some other individual can have 28, you understand? Or 27 or 25, whatever it is, there'll be as many number of bands as possible in the population. So that's why you call it as multi-allelic. Is that okay? And it's multi-locus. Multi-locus means it can be present anywhere. Now coming back to RAPD. RAPD. Now, if you are referring to this, what is this? RAPD is multi-locus or not? RAPD is also multi-locus, right? Because it can bind here, can bind here, can bind here, can bind here, identify polymorphism. So this is also multi-locus. What? Multi-allelic or biallelic? It is only biallelic. Why? Because its presence or absence. Is that okay? Multi-locus, biallelic. Now, what about this? PCR RFLP? We are only targeting a single locus. So this will be single locus. And what? Only one allele you are talking about. It is again biallelic. Is that okay? All of you? Right?
Yes, sir.
Yes.
Okay, guys. Now, so we are talking about single locus biallelic and single locus multi-allelic and all. Okay. These markers are also called as codominant markers. Codominant because you can identify both the alleles. Like whenever you talk about codominant, you're talking about maternal and the paternal side. Maternal and the paternal side. Whenever you're defining it with respect to being codominant. Means, can you identify both of them in an individual? Yes, you can do it. If there are 20 and 20 repeats on both the sides, it's very easy for us to identify it and just mark it and say that, okay, you have got this difference. Whereas, let us say if you have 20 here and you have got 24 here, then what happens? Then it means, then even you can identify the bands and you can say that it is present, both the bands are present, so it's codominant. So mini satellite is a codominant marker, multi-allelic, multi-locus, multi-allelic. Whereas here, this is a codominant single locus biallelic. This is a dominant marker, multi-locus biallelic. That's what you have to understand. Anyways, I'm going to give you a table at the end where everything is defined, but I want you guys to understand what this means, biallelic, multi-allelic, and all this. Okay, now coming to the mini satellite. Sorry, microsatellite. Microsatellite. Okay. Microsatellite markers. Microsatellite markers are from 1 to 6 base pair in length. This is what you need to understand. For the other one, what we talked about 6 to 60 base pair, they are mini satellite markers. That's what we looked at, right? They are mini satellites. So microsatellites are 1 to 6 base pair, and then again tandem repeats here. Now listen to me. Here in microsatellite, what they have done is to make things easy. They are also tandem repeats, but microsatellite markers are normally identified by using a PCR technique. Because let us say this is a region of tandem repeats of the microsatellite. primers are so designed, like from the unique regions adjacent to this particular tandem repeats, right? To amplify this region, So what happens now? So you're targeting a single locus in microsatellites. You're not targeting multi-locus. There, what you are doing, you're taking a DNA, digesting it, and then using a probe. And this is exactly the technique that is used for DNA fingerprinting. And your fingerprint actually matches, your DNA fingerprint actually matches with your parents. You're looking at both the haplotypes coming from the maternal and the paternal side. It has to be correlated with the kind of repeats and the recombination that can take place. Okay, I forgot one important thing, guys. Sorry about that. Okay. I want you guys to refer to one particular thing when you're doing it. Why at all there is a difference of 20 and 24 among individuals? This is mainly because of DNA slippage during recombination. DNA, just read about this, DNA slippage during recombination. We know that during meiosis there is recombination. And what happens? How does recombination occur? Recombination takes place just because of the complementarity. Now, when you look for the complementarity, the sixth repeat here can now match anywhere for the other individual. When the other chromosome is coming, right? You have the other chromosome in the chiasma formation, right? Like this. This sixth can match with the first repeat or the second repeat. And there the segments can exchange. And there arises the difference in the number of repeats. Once again, I'm repeating. The sixth repeat in one chromosome can match with any of the repeats among the 20 chromosomes. And just because of this matching, the recombination, and the segments are exchanged, there'll be a difference in the number of repeats in both the chromosomes. And that's called as DNA slippage. And so why at all there exists a difference between even the individuals of the same family? This is because of DNA slippage. Microsatellites here, we are just talking about that. We are left with 3 minutes. We forgot about the time. Now, you are targeting a single primer. We'll stop it here because I want to discuss something important. Tomorrow I'll continue. There'll be only one class tomorrow, guys. And if